Hello, my name is Catherine Moore, and I'm a senior chemistry major at Lee University, and my presentation is titled From Jellyfish and Anemones to the SMC, the Journey of Two Fluorescent Proteins. In this summer, I worked with my faculty mentor, Dr. Farrell. So before we jump into what my project is about, we have to understand a few concepts first. So the first is, what is fluorescence? So fluorescence occurs when a molecule that contains electrons is excited by a beam of light from an energy source, and this light moves the electrons from a lower excitation level called the ground state to a higher energy level called the excited state. And as it is moving from the excited state to the ground state, so it's dropping in energy level, this is released as a form of light that we are able to see. And the fluorophore is the part of the molecule that is able to re-emit this energy as light. So the first fluorescent protein that we worked with this summer is green fluorescent protein called GFP, and it is derived from the jellyfish species Equirria victoria. And its structure can be seen on the left-hand side of the screen, and it is this rotating beta barrel, and it contains a fluorophore in the center. And that's what you see in the middle image. That's the amino acid sequence of the fluorophore. And then there's an image of the jellyfish on the right-hand side, and the fluorophore is what is responsible for the green that you see within the glowing jellyfish. The other protein that we worked with is called M-cherry, which is a derivative of red fluorescent protein, and it comes from the mushroom anemone species Discosoma. And its fluorophore is also located in the center of the beta barrel that you see on the left-hand side of the screen. So plasmids are small circular DNA that is located within bacterial cells. And there's multiple copies of these located within one cell. And that makes it beneficial to scientists to work with because you're able to extract them from the cell, insert fragments into them, and then place them back in the cell. And there's multiple copies instead of there just being one copy of the chromosomal DNA. So it's a huge advantage for using plasmid DNA. And as you can see in the tube on the right, the chromosomal DNA is stuck onto the side of the tube, and then the plasmid DNA is in the clear part of the liquid. So this summer, we seek to tag, purify, and oxidize those two proteins to allow for covalent dityrosine linkages to form within one protein. So tyrosine is an amino acid. And so we inserted this tag, which contains multiple amino acids, but we made sure it had two tyrosine in it. And then we subjected it to these oxidation conditions, which we'll talk about in a little bit in a second. And whenever we subjected it to this, we wanted to form a covalent dityrosine linkage within one protein. So a previous study has been conducted to show that under these oxidation conditions, that this linkage can form between two or more proteins using horseradish peroxidase, which is an enzyme that catalyzes this reaction, and hydrogen peroxide. And so under these conditions, we're able to take two tyrosine on a tag and link them together covalently. And you can see an image of that in the bottom of the screen. So this is an overview of the process, and then we'll break it down into smaller, more digestible portions in a moment. But the first step is tagging the DNA, which we just talked about a little bit, which is inserting that amino acid sequence. And then we're going to take that tagged DNA and insert it into E. coli cells via the plasmid DNA. Then we're going to sequence the DNA by sending it off and making sure that everything is where it's supposed to be. Then we're going to grow it up and we're going to purify it. We're going to subject it to those oxidizing conditions, and then we're going to use spectroscopy to try to detect dityrosine. So the first step is tagging the DNA. So we designed a primer that includes this tag that we previously discussed, containing the two tyrosine amino acids that we're going to try to get to form dityrosine. And then we were able to use a polymerase chain reaction, which is taking a small amount of DNA and magnifying it by a huge amount. And so we took our tag DNA and used the PCR reaction to magnify it to a larger amount. And then we inserted it into back into the plasmid and transformed it into the E. coli cells. So we just took the plasmid DNA and we manipulated it, added this tag, and then we're going to put it back into the cells. And then we're going to take that product through transformation, which is the process of inserting it back into the cells, and grow it up on these plates to where we see colonies, which there's an image of where you'll actually be able to see some colonies later in the presentation. They look really cool. They're like these little white specks on a plate. It's awesome. So at this point, we have to stop because we're not sure if we inserted the tag correctly. So what we do to ensure that is we send our samples of colonies. So we take a toothpick and pick off a colony and purify the plasmids out of it. We send it off and we make sure, okay, is this what we wanted the amino acid sequence to look like? And is that what is on the plasmids that we purified? So we just make sure that the tag was inserted correctly and that we're ready to move on to the next step. The next step is growing up the tagged protein. So whenever you're growing up the protein, you take a culture tube with the right antibiotic that allows the protein to grow up, and then you let it grow up, and then we induce it with this enzyme that allows the protein to be expressed, and then we let that go for a certain amount of time, and then at this point we will see the protein glowing, which you'll see an image of on the next slide. And then after we see protein glowing, then we purify it using this nickel NTA column, and that is able to hold on to the protein until we use this certain buffer, and then that will wash off the protein. Okay, so on the left-hand side, you can see the process of column purification. So you can see the GFP, this is the protein that we're working with in this step, in the bottom of the column within the beads. So it's that really, really bright highlighted region. That is where our protein is located. This is one of the wash steps. And then on the right, you can see our purified protein after it's gone through this column purification. So this is after it's been grown up and induced and purified, what we're left with. So then at this point, we oxidize our protein, and we're going to add HRP, the horseradish peroxidase, and hydrogen peroxide over 10-minute increments in a water bath, and then we let it sit overnight. So this is the process that a previous study found to work, and so we're basing our study off of this. Okay, so after this point, we should be able to detect dityrosine. To ensure that, we used a fluorometer. So this is a setup of the fluorometer on the right-hand side. So that box, the blue box at the top left-hand corner, is the light source. So the light source travels through that wire into the black box where we load our sample, and then it the other wire is able to track the absorbance of the light and at what wavelength it comes in at. So then it takes that information and goes into the transmitter box, and then that box plugs into a computer. So we're able to see a live feed of the amount of fluorescence of our sample. And so this peak for GFP tells us how well did the protein stay intact. So GFP has an intrinsic fluorescence itself, and dityrosine also has a different fluorescence at different wavelengths. And you'll see a graph of the GFP peak later in the presentation. And so the GFP peak tells us, okay, did the protein stay intact during these oxidation conditions? And the dityrosine peak lets us know if these covalent linkages formed. Okay, so this is the progress that we were able to make with GFP. These are the 3 types of GFP that we worked with. You can see the different tags. Y is the amino acid code for dityrosine. So you can see there are 2 different lengths. And then these are our results. So with the wild type GFP, we see this huge peak for GFP, which is coming in at about 480, 490-ish, somewhere in there. And then you can see this for all 3 types of GFP. So there is the C-tag GFP, and then there is the Y-tag GFP. So this peak is just showing that for all 3 types of GFP, we were able to observe that the protein stayed intact. we were not able to observe a dityrosine peak forming, and that could be for several reasons. We aren't sure if we used a harsh enough condition, oxidizing conditions, to form dityrosine at all. So in the future, we're going to work with manipulating those volumes that we showed on the oxidation slide and increasing them and varying the concentrations of the reagents to see whether or not that will allow dityrosine to form. Okay, so with mCherry this summer, we took many attempts, but we were able to finally tag the protein and grow it up and grow up colonies of it. But we were so we were able to see colonies grow, which was a huge accomplishment. It took a long time to get to that point, but we were not able to obtain a proper sequencing result when we set them off. So they were just in the wrong order when we got it back. So in the future, we're going to work with reworking with the tag that we designed and the primers and making sure that they are in the proper order and redesigning those and trying again to grow up the colonies again and sending it off for sequencing to see if we can get it right this time. So in the future, we're going to work with alternative light sources for the fluorometer to make sure that we are actually able to detect dityrosine with our fluorometer's conditions. And then we will continue working with mCherry by designing a different tag to insert and seeing if that works. And then we will repeat the process in the future with additional fluorescent proteins. So we have 6 different flavors that we can work with in the lab. And those are the ones with the arrow by them. So citrine, M banana, M orange, TD tomato, M plum. Then we've started working with mCherry already, and we're working with GFP still. So we will also be working with various oxidation conditions to see if we can get dityrosine to form and what is the best condition to allow for maximum dityrosine formation while also allowing the protein to stay as intact as possible. So a special shout out to Dr. Farrell. Thank you for your patience with me this summer and for helping me so much, and to the McNair program for funding my summer and giving me the opportunity to do this research. So here are my resources, and if you would like to read further about any of the work that I talked about here, this is a list of my literature review. The previous study conducted is the 3rd bullet point. The protein heteroconjugation article discusses the work with 2 proteins being connected by dityrosine. So thank you for watching. I hope you have a great day.